In this video, I will be going over the use of Faire-seq as a method to identify active regulatory regions in the genome. To understand the method, it's first important to understand chromatin, which is the complex of DNA and proteins called histones. DNA is wound tightly around a core of 8 histones to form a molecular complex called the nucleosome. This forms the structure of chromatin within the nucleus. Chromatin can help control which genes are turned on or off through organization of accessible and inaccessible regions. Think of chromatin like a library of books. When the books are closed on the shelf, the DNA is inactive and genes are not being expressed. Each book is a segment of DNA that can be accessed and used by the cell when needed. When the book is open, the DNA is active and genes are expressed. Faire-seq, which stands for Formaldehyde-Assisted Isolation of Regulatory Elements, is a useful method to help identify the active regulatory regions in the genome. Regulatory elements are non-coding sequences in a genome that control when, where, and how much a gene is expressed. This method is carried out in 4 steps: cross-linking, fragmentation, extraction, and sequencing. In the first step, cells are treated with formaldehyde, which forms covalent bonds between proteins and DNA. This chemical fixation will lock the proteins and DNA at their location in the genome and is more efficient in closed chromatin than it is in open chromatin. In the second step, the cross-linked chromatin is broken apart through a process of sonication, which uses high-frequency sound waves to create microscopic bubbles that collapse and rupture the cell membrane. This breaks the DNA into smaller fragments in a process called DNA shearing. The third step involves phenylchloroform extraction. The DNA sample is mixed with phenyl and chloroform. Phenyl denatures and precipitates proteins, while chloroform helps separate the phases. After centrifugation, the mixture separates into 2 distinct layers based on density. The organic layer is the denser lower layer containing phenol and chloroform. It traps proteins and lipids and contains the protein-bound closed chromatin. The aqueous layer is the lighter upper layer containing water and dissolved substances. Because DNA is a polar molecule, it stays dissolved in the aqueous, water-based phase. The top aqueous layer is carefully transferred to a new tube, leaving the organic layer and protein-bound DNA behind. This process is repeated several times to increase the purity of the sample. The cross-linked DNA, also known as background DNA, that is in the organic phase, is reverse cross-linked and solubilized to use as a control that can be compared to the DNA from the aqueous phase. The final step is DNA sequencing. The DNA from the aqueous phase, which is the open chromatin, is then prepared for high-throughput next-generation sequencing. This is a useful technology that can sequence millions to billions of DNA fragments simultaneously. Once sequenced, the locations of the active regulatory regions are revealed. Sequencing reads are mapped to a reference genome, which is a standardized DNA baseline that's built from multiple individuals of the same species. This can allow researchers to identify regions of significance that correspond to open chromatin. In conclusion, Faire-seq is a useful method to determine sequences of DNA regions in the genome associated with regulatory activities. What makes this method unique is that it is based on differences in cross-linking efficiencies between DNA and nucleosomes or sequence-specific binding proteins. This method also does not require antibodies or enzymes and can be used to analyze any type of cell.